SCP-047 was recovered only after a full compromise at site. Its first uncontrolled release triggered a microbial bloom that killed every worker in a secure laboratory. Now, the object sits inside a hermetically sealed box, locked in storage locker 47A within P3 Secure Biohazard Lab 047B. Exposure means quarantine, senior authorization, and no casual SCP-047 is a breached, heavily rusted gas cylinder made from an iron-tracted alloy. In open air, the cylinder slowly evaporates and sheds a mutagenic gas. The gas does nothing to human cells. Instead, it alters prokaryotes, especially the bacteria that already live on skin and inside the body, turning ordinary commensals into SCP-047-1 superbugs. Across species, the mutations keep the same advantages: environmental survivability, full antibiotic resistance, faster reproduction, gram-positive sporulation, gram-negative plasmid exchange, and broader transmission.
Although SCP-047-1 kills through mundane biological pathways, the process works far better on bacteria living on a human host than in culture.
After the breach of 30-01 to the home zero, mutating already pathogenic species was banned, and existing samples had to be destroyed. Proparanobacterium 047. It was documented in D1V978. 2 days after exposure, it colonizes sebaceous glands, shifts skin pH to toxic levels, and breaks down the skin itself.
This strain moves through skin-to-skin contact and can stay active on inorganic surfaces for 5 hours. Visible symptoms can appear within hours. Mortality reaches roughly 40%, and dead victims are to be incinerated. Streptococcus 047-C begins with inflammation in the mouth and esophagus, then opens sores that let the infection into the bloodstream. Death usually comes through septic spread and infectious endocarditis. Unlike the contact-driven skin strain, a 047-C spreads by droplets and can remain active indefinitely through sporulation. Mortality is about 35%, and survivors may be left with a recurring chronic condition. Clostridium 047-A was developed from tissue culture and has never been exposed to a human subject. No remaining samples are under Foundation control, which leaves even its basic pathogenic profile unresolved. Transmission is presumed fecal, but its smaller spores may aerosolize with flatus, and inhalation outcomes cannot be predicted, because the strain could destroy the intestinal lining. Corpses are not to be incinerated before etiological study. Testing logs say researchers were trying to contain data expunged inside a class pressure cylinder when combined reactions produced SCP-047. The resulting breach transformed the vessel itself into a biohazard source. Once the damaged cylinder vented, uncounted SCP-047-1 species bloomed through the laboratory microbiota and killed the entire staff within hours. Standard Foundation quarantine then contained the outbreak before it escaped the site. Every entry into Lab 047B is recorded. Exposure is treated as a Priority 4 contagious biohazard, and project manager approval plus at least 105 clearance are required. If SCP-047-1 escapes the lab, personnel go to Quarantine Suite Q-047 and Lockdown Protocol 047-01 Yersinia begins. SCP-047 remains Keter because a small rusted cylinder can turn ordinary human microbiota into fast-spreading resistant pathogens. The object is contained only so long as the seal holds, the rules are obeyed, and no one forgets what open air does.